Hello everyone. In this video we're going to talk about how cells differentiate into different types of cells in an organism and eventually form an organism that has a 3D shape. So we finished talking about apoptosis in class. So we talked about blooding, programmed cell death, and how apoptosis also happens in your body to protect you from damaged cells. And that apoptosis kind of tells you about common ancestry as well, because many eukaryotic organisms has the same mechanism of apoptosis, just like we do. So the next one that we want to talk about is this picture right here. I told you in class that in order to form different digits of your hand, this process requires apoptosis during the developmental process of an embryo. So as you can see right here, earlier in the developmental process of an embryo, we have this webbed hand, right? You can kind of see the outline of the fingers, and you can kind of see bones forming inside the hand, but really there's not, and there's no finger yet. There aren't any fingers yet, right? So as you can see, as time goes on, apoptosis happens in between these digits to eventually allow you to have fingers. So apoptosis is also essential for the normal development of the nervous system, as is said right here, and is important for the morphogenesis of hands and feet. So remember, morphogenesis is the formation of kind of your 3D body shape and every part of your body, your tissues and cells, well, your tissues and organs and organ systems. So pattern formation is the setting up of the body plan. It's developmental of a spatial organization of tissues and organs in an organism, right? Where do we have a certain type of tissue? Where are muscle tissues supposed to be in, right? You're supposed to have muscle tissues in the muscles, not right on your face. And how is that process controlled? Pattern formation discusses how this pattern happens, right? And then we have the organs, right? The organ's supposed to be inside your body, right? Inside, right here somewhere. But some people are born with the heart outside of their bodies, for example. So that's a problem. So that there might be pattern formation problems that's allowing this organ to be kind of, be at the wrong place of the part of the body, right? But usually you wouldn't have hearts developing on your hand, because first off, that wouldn't work. And second off, your body, your cells send signals to each other so that you don't grow a heart right in your palm. And then positional formation is kind of what we talk about, the anterior posterior. So you should know that anterior is talking about the head region. Posterior is talking about the tail. So for us, it's kind of, the feet and the lower part of your body. Dorsal, ventral, you should also know these two words. Dorsal means the back. So we have a back. Ventral means the belly part. So for a lot of, insects, other eukaryotic organisms, lizards, they all have a dorsal, ventral, anterior, posterior. But we also have right and left axis, right? So if we look at a human, we have an axis, and the left and right look pretty much the same on the outside, but your organs are placed at different places, right? So your heart is on this side of your body, right? And you wouldn't have one on the other side, and you wouldn't have one in your tummy. And how that's oriented, left or right, is also dependent on the pattern formation. Here is the cause of the pattern formation, right? So we're talking about how we have this anterior, posterior, dorsal, ventral, left and right. But what causes this? The same two things that we've talked about before. First one is called cytoplasmic determinants, right? Remember, we have the egg cell, there's cytoplasm inside the egg cell that has uneven distribution of certain chemicals, including mRNA and proteins. And these, this one egg cell split in 2 cells, and then 4 cells, and then 8 cells, and eventually 32 cells and more. These cytoplasmic determinant is going to determine what these cells are going to kind of become, right? What path are they on? Are they on the path to become the head? Are they on the path to become the tail? So that's cytoplasmic determinant. And then we have inductive signals, which is when the determination process of a cell and signals sent between cells and within a cell. So here we have uneven distribution of morphogens. So we'll talk about that, and then controlled by homeotic genes. So you should know that homeotic genes help determining cell fate. So we'll talk about that in a second. So the next one is, if you were to look at this picture right here, we have a fruit fly as what we call a model organism. So make sure you know what a model organism is and how we determine something is good for to be a model organism. So fruit fly is a typical model organism that scientists in the entire world use because they're small, they reproduce fast, and we know, we came to learn a lot about them. So fruit flies are a type of arthropods. Then they have a structure that have the anterior, posterior axis, dorsal, ventral, and right and left. And cytoplasmic determinants in the unfertilized egg determine the axis before fertilization even happens. So isn't that crazy? There's one cell, this egg cell, that's not even fertilized. The anterior, posterior, dorsal, ventral, left, and right of this future fruit fly is already predetermined. This fruit fly is not going to become a ball of cell because of the cytoplasmic determinants way before this fruit fly is even a fruit fly. So I think that's pretty cool. So if you were to look at this picture, it kind of walks you through how this process works. We start with this developing egg. As you can see, we have the nucleus of the egg and we have the cytoplasm of the egg. And we also have some surrounding cells that are kind of act as supports, or what we call it nurse cells. So these nurse cells or follicle cells help the formation of the egg when it matures. So these nurse cells contain a lot of nutrients, a lot of proteins that are sent to the actual egg part, which is right here. And then as you see, as time goes on, the egg cell becomes bigger and bigger, and it takes nutrients from the nurse cell, and the nurse cells become depleted. And these follicle cells are kind of become the outside shell of the egg. So when a lot of organisms such as fruit flies, when they lay their eggs, or they're not like the eggs that you eat that has a hard shell, right? These are, individual eggs that they lay on the medium that you contain them. I'll show you this in class if you want to look at them. They're pretty cool. And these eggs will eventually turn into fruit flies. And before there are fruit flies, and then you can actually see them in our lab in these vials with fruit flies, you can see these developing embryo that will become these little tiny worms before they can start flying. So, but before that happens, this egg cell is going to get fertilized and then become segmented embryo. So as you can see here, we already have the head and the tail orientation, and this whole orientation of head and tail and back and front and left and right is already determined by the uneven distribution of chemicals inside this egg cell, which is called cytoplasmic determinants. I forgot about it for a second, I'm sorry. So this shows you how this works for one fruit fly. And this is already way more cells than one fertilized egg, even though they're the same size, so that's easier for you to see. But this is a lot of cells. This is a segmented embryo already. And now we have this larva, head and tail. So there's this guy, Lewis, and these other guys. These scientists won a Nobel Prize in 1995, which you weren't born yet. I was 2 years old. They kind of decoded the entire pattern formation in Drosophila. So they kind of tracked each cell and what is the developmental process of each cell, right? Well, we have one cell split into two cells and then four cells, but how does each cell become the later cells, and what role do they play in the organism, and where do they locate in the organism? So they kind of study that. And Lewis right here discovered something called the homeotic genes. The homeotic genes control pattern formation in late embryo, larva, and adult stage, right? So these homeotic genes kind of tells a fruit fly, let's say I'm a fruit fly and I fly. The homeotic genes over here in the fruit fly larva as the embryo keeps on developing, these homeotic genes are going to send signals to nearby cells and to the cell itself to allow an arm to grow right here, right? Same thing over here. There are homeotic genes in charge of telling everybody that it's arms and cells that are in charge of growing arms supposed to supposed to turn into arm cells. Right here, obviously, there are many different types of cells in an arm, even for a fruit fly, but you get the idea. So that's homeotic genes. It's really important. So this right here shows you what happens if homeotic genes are expressed incorrectly. So normally you would have eyes where they're supposed to be, the antenna grown right here, because the homeotic genes tell them that this is the place to grow, right? And also the cytoplasmic determinants tells the fruit fly that this is the location out of this entire embryo. This is the place to grow ahead. And however, if the homeotic gene that's in control of growing a leg gets expressed where the head is, then it's going to grow a leg instead of antenna on the head, right? So as you can see, there's leg growing from the head, and this is not great for the fruit fly, but it's great for us scientists, or you scientists, maybe one day when you become one, that homeotic genes is in charge of telling the entire body how the pattern's supposed to form, right? The body pattern, okay? And we think that these homeotic genes and the mutation of these homeotic genes is what lead to a lot of the evolution. So for example, I told this to the blue class, but we didn't talk about it in orange class, so I'll say it one more time. So let's say, I'm just making this up, but this whole idea is not made up, okay? So let's say we have a fruit fly that's supposed to have, I don't even know how many legs they have. I think they have 6 or 8. Anyway, so let's say they have 6 or 8 because the homeotic genes tells the fruit fly that these are the places to grow legs. And then, mutations happen that allow these fruit flies to grow even more legs. And somehow the environment determined that having even more legs than the original normal fruit flies, the wild-type fruit fly, is more fit for the environment. So that will allow these fruit flies with more legs to evolve, right? Because they're more fit for the environment. So those are survival of the fittest. They're fit. So then the result is, after, let's say, 100 years, we'll have fruit flies with more legs, right? I just made this up. Obviously, the timeline doesn't work like this, and not every organism's development of legs and arms and heads works like this, but this is something that the mutation of the homeotic genes is something that adds on to the variations within a population. And obviously, you have to look at a very large population. We don't get people with 3 arms, very often. At least, I don't know if that's the thing. You could look it up. So here we have, so these 2 scientists, I don't dare to say their names. These 2 scientists actually identified all the genes that affect segment formation in Drosophila, right? So Drosophila have 14,000 genes, and out of those 14,000 genes, there's about 1,300, 1,400 genes that are specifically used for pattern formation. Those genes are the homeotic genes, or they're the genes that determine the pattern of the fruit fly. Where are wings supposed to be growing? Where are eye cells supposed to be made? Right? Where are cells for the tail? Do they have a tail? Where are cells for the legs supposed to be, specialized? Right? All those specialization or differentiation of the cells, and where those differentiations supposed to happen. Those are the genes that we're talking about right here in this bullet point. And then many of the identified mutations were embryonic lethals, causing death during embryogenesis. What that means is that because these, I think 1,300, 1,400 genes are so important, they're so important because that's what gives you the organism, right? Because if you have a ball of cells, that's usually not a living organism. If you're trying to make a fruit fly, this fruit fly has to at least have the fruit fly shape in order to live. So if you have mutations in these crucial homeotic genes that determine the body pattern, then it's often embryonic lethal, which means the embryo itself will die, and this fruit fly will not even be born. So a miscarriage will happen. I don't know if that's what you call it in fruit flies, but you get the idea. Oh wait, I completely messed up the numbers. It's 120, not 1300. I'm sorry. So it's 120 genes that they found that are essential for the normal segmentation, right? But if you think about it, out of all those genes, being able to identify specifically 120 genes, that takes a lot of work. And you might say, why does this help us at all, right? But because this understanding of how these genes work, first off, we all have a common ancestor, so probably some of these 120 genes that are expressed in Drosophila that have a certain function for normal segmentation or pattern formation for fruit flies might be common or very similar to what we have. And there's also, being able to understand how each cell come about, and, just point at this cell right here, this one cell that I can't even point, right? This cell, where was the cell before that came from? Where was the cell before that come from, right? So if you can trace the cells all the way back to its origin and how they come about, it can help you understand a lot of the potential diseases and, how does the cell accumulate its mutation, for example, right? So that's really what the studying of these model organisms is for. It might seem a little boring to you at the time, but really if you come to appreciate how much, the small amount of understanding that you gain for this one model organism, how much that can help the entire humanity, in the future, the collective effort and intelligence really can make a big difference. So that's my, if you become a scientist or a doctor in the future, I hope you can appreciate those Drosophila. I certainly in the past, back in college, did not appreciate Drosophila very much, and they're kind of annoying flying around in my dorm sometimes, but they've definitely contributed a lot to science. All right, axis establishment. Here we have maternal effect, something called maternal effect genes and egg polarity genes. They mean the same thing. So what those are the genes that encode cytoplasmic determinants that initially establish the axis of the body of Drosophila, or us, right? If you remember, we talked about cytoplasmic determinant, right? What are the genes that actually code for those proteins that are unevenly distributed in the egg cell? These maternal effect genes and egg polarity genes are the ones. If you think about it, right, these are the genes that affect the entire orientation of the axis of an organism's body. So if you have mutations in those genes, there is going to be a big problem, right? So you will definitely see a resulting phenotype if the offspring is born at all is going to be quite mutated. And then there's also the part where most of the time these mutations are lethal, because if you mess up the entire, if you mess up the axis of the body, who knows where grows what, and it's not going to work, right? So it's like if you were to take a Play-Doh and you're trying to make whatever it is that you're trying to make, but you put the head at the wrong place, and where you get two heads, that's creative, but in real life it doesn't work so well. So as I said, embryonic lethal. That's one. So here's an example of how this maternal effect gene works. Okay, so this gene is called bicoid. This is a maternal effect gene which affects the front half of the body. So if an embryo has no functional bicoid gene, it will lack the front half of his body, and it will have posterior structures at both ends. So what does that mean for a human, right? If this were to actually happen to a human, which I suppose it's so embryonic lethal that it will never happen, because a human cannot operate without a brain, and your the mother's body will know that there's something seriously wrong with this baby before this baby even go on to the later part of the embryonic development. But how does this look like for a fruit fly? When the bicoid gene is missing, you would have, instead of a head and a tail in a fruit fly embryo, you would have a tail and a tail. All right. So this is actually a larva. It's not just an embryo. This is a larva. So they're kind of like little tiny caterpillars, but they're called larvae. And they're not caterpillars, but they kind of look like one, and they it does this thing. Right? Yeah, they walk like that. And you wouldn't call it walk. Anyway, so this is really wrong, right? This fruit fly does not have a head. It has two tails. This is not going to work out. This fruit fly is not going to survive. But luckily for science, this tells us how important it is to have the correct maternal effect genes, or egg polarity genes. So now that I think about this, I'm just letting my brain take it away. Don't listen to me, but I'm just thinking, right? So I wonder if this maternal effect gene could be the cause of some infertility in people, because if you were to have a messed up, if a person were to have a messed up maternal effect gene, then this embryo is not gonna live. So I don't know. You should look this up. Maybe I'll look this up later. All right, the next one is called morphogen gradient hypothesis. Let's break this down, right? Gradient is the distribution of something. Hypothesis is hypothesis. Morphogen is chemicals that, well, what is morphogens? Where'd it go? Morphogens help set up the embryo's axes and other features. So those are kind of chemicals that get sent throughout an organism's body. We'll see pictures of that. It will make a lot more sense. So this is not, so we had this maternal, where'd it go? So we have the maternal effect gene, which affects the distribution of molecules inside the egg cell, right? So the morphogen is kind of similar to that, but except it's not in one egg cell now, it's talking about the entire embryo. So this is chemicals that get sent throughout the entire embryo and kind of help determining the embryo's axis as well. So this is another one that happens after the embryo is already formed a little bit. And the maternal effect gene and the maternal effect gene acts on the embryo before it's even an embryo. So it acts on the unfertilized egg. Okay, so kind of wrap your head around that. So this right here, the bicoid. Okay, I don't know why I jumped back to bicoid. So the bicoid mRNA is highly concentrated at the anterior end of the embryo, and then after the egg is fertilized, the mRNA is translated into bicoid protein, which diffuses from the anterior end to the other end. So the result is a gradient of bicoid proteins. Okay, so let's see. So you would call this maternal effect gene, right, which comes from the mother, but then this gene also it comes from the mother, and it has an uneven distribution inside the original egg. But then, as the organism, as the embryo develops, it's also going to affect the entire embryo. So how does that actually look like? Should be on the next page. So this right here, you should watch this video because you can't hear the sound on mine. So definitely go watch this video. It's going to show you how this works. But this picture also shows you how this works. We have the bicoid mRNA in the matured unfertilized egg, right? This is the original egg that has the uneven distribution of the mRNA. And then once it's fertilized, and this fertilized egg become an early embryo, which also takes a similar shape, but this is one cell, this is many, many cells. And what happens is this mRNA is going to make proteins that get distributed in the early embryo as well. So as you can see, we have a higher concentration in the anterior end, and then a lower concentration of this bicoid protein in the posterior end and in the middle of the body, right? So at the beginning of life of this unfertilized egg, as well as early embryo, the uneven distribution of chemicals, whether it's mRNA or protein, is going to help determine the axis of the body. So the last thing that we want to talk about was this bicoid thing. This bicoid forms the head, right? So what we also know is that if we were to inject the bicoid mRNA into a certain region of the embryo, a head is going to form. It's not necessarily functional. In fact, it's not going to be a functional head, but it's going to be, cells that are formed in the head being formed there. You're not going to have 5 functional heads on a fruit fly, but there are going to be head-looking structures, at least. So make sure you watch this video. It's not very long. All right. So this research is very important for 3 different reasons. First off, it helps us identify a specific protein required for some early steps in pattern formation, right? Which is also useful for the understanding of how the pattern formation is done in human beings, because that's what we all care about, human beings, unfortunately sometimes. And it helps increasing the understanding of the mother's critical role in embryo development, right? Again, like I said in class, it's not just the offspring DNA, which comes from the parent's DNA, right? But it's not just the DNA that determines how the embryo develops. The mother's, even just the cytoplasm of the egg cell, which is not the DNA of the embryo, has a huge effect on how the embryo develops. And obviously the environment that's set up in an organism's body, and the hormones and all that, all of those have an effect on the embryonic development. There's also, what is this? The gradients of morphogens. So those are chemicals within an embryo with many cells, and the distribution of these chemicals, right? That can determine the polarity and position in the embryo. So we have the cytoplasmic determinants. So those are kind of the earlier step of determining the axis of the body. And then, sorry about the hair. But then the morphogens, the distribution of morphogen, this is kind of for a little bit later on, not exactly, it's not late during embryonic development, but at least we have an embryo now. It's not just the egg cell. So moving on to cloning. So we're not going to talk about this a whole lot because we have some classmates already talking about cloning last week, but just a few things that I want to mention. What exactly is cloning? We're using one or more organisms, or we can develop one or more organisms from a single cell without meiosis or fertilization. If you can remember back, meiosis, review time, right? Meiosis produces gametes that are haploid. And if you put 2 gametes together, one maternal gamete, one paternal gamete, if you put those 2 together and form a zygote, this process is called fertilization. That's how organisms go through sexual reproduction and produce new multicellular organisms. However, with cloning, we can have individuals that are genetically identical to one parent that donated a single cell, right? So this doesn't have to be a male or a female. This could be anything, because all it's donating is the nucleus. And this kind of help us, so cloning, obviously we're not going to clone a human, at least anytime soon, but there's the part that we can clone some stem cells. So